running here. Okay. And share screen. How is that looking? Okay, great. Well, yeah, it's such an honor to speak in this forum. Thank you again. So, broadly speaking, if I'm new to you, what I've been doing, what my lab's been doing, is broadly trying to understand basic large-scale brain mechanisms of human cognition in health and disease, and to see if we can help inform the development of drug-free neuroscience therapeutics for people with cognitive brain disorders. So I'm sure I don't have to tell this audience that cognitive brain disorders are some of the most disabling brain disorders, most disabling health states in the world. So these are disorders like schizophrenia, Alzheimer's disease, obsessive-compulsive disorder. And so for these disorders, existing drug, surgical, and behavioral therapies are quite limited. So I'm sure, as you know, they're limited. They have a number of limitations associated with them, such as heterogeneous treatment outcomes. They're limited by the slow time course of symptom resolution. They're limited by accompanying risks and side effects. And so as a result, we think that there is this growing need for alternative approaches, for new, potentially personalizable therapeutic interventions that could potentially be fast acting and long lasting and help people with cognitive brain disorders. So for this, non-invasive neuromodulation technology is an emerging class of tools that offers translational potential for neurocognitive disorders. So these are tools like transcranial electrical stimulation, transcranial magnetic stimulation, transcranial ultrasound stimulation. Within the transcranial electrical stimulation class, there's a tool called high definition transcranial alternating current stimulation, or HDTACS. And this is a tool that we've been using quite a lot in my lab since I got to BU. So it allows us to potentially use the importance of large-scale rhythmic brain activity patterns, and it promises really unprecedented control over augmenting the rhythmic activity patterns of these large-scale brain networks that are implicated in cognitive function and dysfunction. And so in this talk, the plan is to share some of my lab's latest research in what might be called precision or personalizable HDTACS, which has shown some promise in being able to both non-invasively and safely manipulate neural population dynamics, these large-scale brain activities, and improve components of human cognition, and do so also in a selective, potentially long-lasting manner. So the hope is that this research might contribute to setting basic science groundwork for the future development of like a network-based therapeutic that's drug-free, which could potentially, I imagine, open up an entirely new realm of treatment options for patients with a variety of different brain disorders. At least that's the most optimistic view. So the approach that we take to brain stimulation or neuromodulation design is really grounded in decades of fundamental neuroscience that views cognition as arising from synchronous electrophysiological oscillations or rhythms across different spatial and temporal scales. So if the concept of a brain rhythm or oscillation is new.
This is not Joanne. Yeah. Okay.
If you're Mary Beth, could you please mute yourself?
So if you're new to the idea of an oscillation or brain rhythm, it's worth pointing out that these are rhythmic or repetitive patterns of brain activity. So brain rhythm comes from inhibitory and excitatory postsynaptic fluctuations that are evident in the cyclic changes of local field potentials, electrocorticography, electroencephalogram, magnetoencephalogram. And so there are postsynaptic fluctuations. But more importantly, we ask, like, well, what do oscillations do? And so functionally, when you have rhythms that are temporally correlated, they're correlated in time or they're synchronized, they could potentially sculpt or coordinate neurophysiological dynamics. That is, they can sculpt or shape spiking activity, or in the coin of the realm in neuroscience for representing information in the brain, right? And so the goal of oscillations then is to facilitate precise but flexible communication in the brain. That's important for cognition and goal-directed behavior. So you can imagine synchronized brain rhythms as representing a kind of gate or an excitability window in the brain. So when there's synchronization, the gate is open, and information in the form of spikes of action potentials can flow more readily within and between brain regions. And obviously, when there's a decrease in synchronization, the gate is closed, the excitability is closed, so it's less likely to be this flow of information. In addition to coordinating spike timing, neural synchronization can also increase the likelihood of inducing spike timing dependent plasticity. So it can induce spike timing dependent plasticity, and as a result promote flexible cognitive functions. And it's because of this kind of dual mechanistic role of information transmission and plasticity that neural synchronization, broadly construed, is recognized as a really basic neurophysiological process important for human cognition and flexible, intelligent behavior. What's exciting now is that we have a tool that can potentially isolate and act on, in a non-invasive way, these patterns of neural synchronization in humans. So the synchrony of neural rhythms within and between cortical regions can be potentially non-invasively changed using forms of rhythmic neuromodulation, like TACS, transcranial alternating current stimulation. And so in this technique, low intensity alternating currents are delivered to the scalp via electrodes, with the goal of inducing an alternating electric field gradients in a targeted brain region. And these alternating electric fields have been shown to manipulate spike timing or spike phase without changing spike rate, which is interesting. And that's what I'm showing here at the bottom of the left panel in the bottom of the slide. You see entrainment of the spikes on the TACS condition, and trained to the frequency of the alternating current applied extracranially, in contrast to the sham condition where you see more scattered spikes over time. That's been replicated in this other paper here by Johnson, but they went further and showed this parametric manipulation. So you can parametrically, you can increase the magnitude of the entrainment of the individual spikes in these non-human primates by stimulating extracranially with the same intensities that we do in humans, and do so in a parametric way. So as you increase the intensity of the TACS from 0.5 to 1 to 1.5 milliamp intensity, you can see a corresponding enhancement in spike phase, again, without changing spike rate. And the entrainment of neuronal activity can lead to the induction of neuroplasticity, which allows for any neurobehavioral changes to persist beyond the duration of neuromodulation. In a nutshell, these are the two mechanisms of action that we think are underlying the effects of TACS. both entrainment and neuroplasticity. So there have been some notable advances in TACS technology and protocol design in the last several years that really offer unprecedented functional anatomical targeting over and above that of conventional TACS that we also employ in the lab. So in the conventional approach, you can see here the application of these large sponge pads is common, maybe 5 by 7 centimeters, which induces an electric field that really makes contact with large swaths of cortex. The spatial scale here, spatial resolution, is on the order of lobes. Whereas in the high resolution approach that we use, you have substantially smaller electrodes, these often 12 millimeter diameter ring electrodes, which you can configure on the scalp in a center surround sink source manner. So here, where you have that red anode flanked by 4 cathodes, allowing you to really funnel the current and increase the spatial resolution, the focality to a target brain region. So there's been significant enhancement in spatial resolution in this space. In addition, you can see we're using computational models of electric fields to help guide the creation of these montages. So a TACS design, what we mean by that is, where are you placing the electrodes? How many electrodes are you using? What is the intensity at each electrode? And again, it's guided by these models of electric fields, which further improves the focality of the stimulation. And this enhanced focality, this enhanced spatial resolution with modeling also minimizes the likelihood of producing unwanted effects in other anatomical structures like the retina and extracranial tissue. We can also now target, with this high resolution approach with HD-TACS, we can target more than one brain region simultaneously, which opens up a lot of new questions that can be addressed. So we can target more than one brain area at a time. And as a result, we can try to augment the communication, and in particular, the phase-based connectivity, which I want to talk more about during this talk, between 2 brain areas. That is, we can sort of play with the timing of the brain interaction between regions that are quite far apart, these really spatially segregated cortical regions. So the HD-TACS can be used, again, not only to non-invasively manipulate brain rhythms in a frequency-specific manner, and not only to do it in a single region like conventional TACS, but also potentially to modulate these streams of connectivity, these streams of phase-based connectivity over long distances. Recently, there's been another push toward personalization that's captured the attention of a lot of brain stimulation researchers and others in neighboring fields. There's been really intense attention devoted, and with personalization, key properties of individual neurophysiology and individual neuroanatomy, such as the intrinsic rhythmic frequency of a neuronal circuit, are taken into consideration when designing neuromodulation protocols that tailor to an individual. And so using a combination of these developments in TACS technology and protocol design, my group has shown that it's possible to functionally, to get really functionally specific, bidirectional, can push circuits up and down, and relatively long lasting, in some cases, control over network synchronization and the behaviors that they support. And so this work that we're doing now falls roughly into these 3 categories. So these are the active research streams in the lab. They have to do with memory deficits in aging and Alzheimer's disease, executive function and dysfunction in schizophrenia, and reward processing and decision-making in people with anxiety and related disorders, in particular, obsessive-compulsive disorder. And again, the big goal of these research programs is to try to bring together the latest and greatest in non-invasive neuromodulation technology to create personalized, high resolution designs that can better steer plasticity mechanisms in human cognition, so we can better understand and potentially rectify neurodegenerative, neuropsychiatric disorders. So my plan today is to talk about the memory and aging research area in the lab, but of course I'm happy to talk about any of these threads with you. So aging. So as we grow older, we often grow wiser. Hopefully that's the case. And it's true that according to cross-sectional longitudinal studies, there's a variety of mental faculties that remain relatively preserved and stable across the adult lifespan. So these are abilities like autobiographical memory, emotional processing, automatic memory processes, theory of mind performance, the vocabulary and semantic knowledge. All of these functions are kind of remarkably preserved and stable across the adult lifespan. On the other hand, we do experience memory loss and cognitive decline, right? Kind of all experience this as we age, that interferes to a greater or lesser extent with our activities of daily living. So this is just a summary slide from seminal work by Park and colleagues showing this robust linear decline in visual, spatial, and verbal memory loss across the adult life. And there's decades of research now showing similar results from cross-sectional longitudinal studies that find these robust, rather linear declines in numerous cognitive abilities, such as working memory and executive processes. So here are just some examples of the linear decline in various working memory tasks across different sensory modalities. And this is going to be the focus for this talk, and my lab has a particularly keen interest in working memory. Of course, it's kind of fun to take a step back and just appreciate the power of kind of cognitive tools that we have at our disposal as human beings and the role of working memory in them, right? They give rise to these exceptional accomplishments that we make, developing life-saving vaccines, exploring explorations to the moon, establishing civilizations. But our brains can store seemingly just infinite amount of information that we've learned over a lifetime, and then use that acquired knowledge to construct a coherent sense of the world around us. On the other hand, despite these remarkable capabilities, the human cognitive system is also dramatically restricted in how much information it can actively hold on to at any given moment. That is, like, despite the general feeling that we have this this awareness of much of our immediate environment, we have access to, in reality, only a tiny little sliver of it, sampling the physical world around us in these little bite-sized amounts of information for only 3 to 4 objects at a time. And so this working memory system allows us to temporarily hold behaviorally useful information in the mind that's readily accessible in a readily accessible state, so we can then use it to perform complex cognitive tasks like reasoning and problem solving. And so it's a capacity where we work on information, on representations, to comprehend language, perform mathematical calculations, a lot of things. And it's often because of that, psychologists have conceptualized it as a work pad or a sketch pad, a workbench of the mind. And so it's this really fundamental building block of higher order information processing. And we're still learning about it. We haven't solved all the questions here. In fact, some of the most basic questions in neuroscience are still quite a mystery. That is, for example, how does the brain represent information and carry that information forward in time? Believe it or not, that's still incredibly mysterious. So working memory has been divided into these often 3 processing stages of encoding, maintenance, and retrieval. Much work has focused on the maintenance stage, that holding on to the information. And there's been a leading theory originally proposed by Donald Hebb in 1949 for how we hold information and carry it forward in time. And this may be accomplished, head thought, through reverberating activity in distributed cell assemblies. So here's a schematic of a Hebbian cell assembly here with the arrows and the numbers. And the arrows represent a sequence of a neural cell assembly where each cell assembly fires according to the number on the top of each arrow. And once each cell assembly in this loop has fired, the sequence begins again with the first cell assembly. And so there's this sequence of reactivating cell assemblies that creates a loop, a dynamic loop of activity, a reverberating circuit, which, as the theory proposes, could potentially support sustained neural firing. Sustained neural firing is like the canonical neural representation of working memory. You hold something in mind, you famously, like Goldman Rakesh and others, have shown that you have this single units that burst and then sustain this burst, this high burst rate through delay periods in working memory tasks. So let's look at some of the kind of famous examples for this proposal. So here's a delayed match ocular motor delayed ocular motor response task that you see in lots of monkey work as well as human work. And in this task, you fixate a central point. There's a peripheral target that appears. It goes away. You store that target in your mind briefly, and then you're cued to make a saccadic eye movement to remembered location, and then you get a juice reward or water reward. And in contexts like this, what's commonly observed is spike field coherence in the gamma band. So this measures spike field coherence. What it shows is that there's a temporal alignment between the activity of spiking neurons in distributed areas and the phase of the local field potential. In this case, in a gamma frequency range, roughly 50 hertz.
Whoa. Hi. How are you? I am good. I'm watching.
Liz, you should mute yourself. We can hear you.
And so another fine example that provides some support to the Hebbian cell assembly idea for working memory is here in hippocampus. So here's this other example, but it shows the presence of low frequency activity, the importance of it. And so here you get spiking activity, pools of cells that lock to the trough of a theta frequency, 4 to 8 hertz in the hippocampus. And you can see how it's organizing these pools of cells, different distributed pools of cells locking on to the trough of this low frequency local field potential. It's another case of this spike field coherence. So these are examples of observations where that like rhythmic activity, whether it's low frequency, theta and alpha, whether it's high frequency like beta or gamma, that they're able to kind of come together and form these cohesive organized groups of neurons by establishing these transient temporal correlations, by being synchronized, in this case across spatial scales from fields to spikes. Well, we don't work there with humans. That's a lot of systems neuroscience, and there's a beautiful rich body of systems neuroscience with animal models that we ride on the heels of, that really lays the basis for a lot of the theorizing and experimental work that we do in human cognitive neuroscience. So it's been proposed if we pop up to the cognitive neuroscience level, sulci and gyri and whole brain scalp electrophysiology and imaging and so on, it's been proposed that synchronization at this high level of macroscopic scale, millions of cells interacting in concert, that these fast temporal correlations, this form of synchronization between brain rhythms of different frequencies, may serve as a mechanism for online working memory storage. In particular, there's this curious signature called theta gamma phase amplitude coupling. And this particular electrophysiological signature is where the amplitude of a fast wave like gamma is coupled or correlated to a phase interval of a slow wave like theta. So you can see this fast wave in the bottom, it peaks where also the peak of this slower wave on top is. When that occurs in a regular manner, you get phase amplitude coupling between theta and gamma in this case. And so this has been observed during in the maintenance of working memory tasks in animals and humans. And I'll give you an example here of a pre-surgical epileptic patient with an electrode advanced down in temporal cortex. And you can see just as this person loads objects into their working memory, you can see this parametric increase in the magnitude of this signature, this theta gamma phase amplitude coupling. The pack effect phase amplitude coupling it's usually observed in like temporal lobe structures, these sensory processing areas of the brain. And again, it's thought to be a neural correlate of working memory storage. But in addition to the temporal lobes, areas of prefrontal cortex are also known to play a role in working memory. And the more current theories propose that the prefrontal cortex is not playing a role so much in storage, but in top-down control and monitoring of those objects being stored in posterior regions. And one proposal for how the prefrontal region interacts with those sensory regions way back in the back of the head is through phase synchronization. Phase synchronization is another kind of neural coding motif. Phase amplitude coupling is one coding scheme, and phase synchronization is another coding scheme. And this is when two oscillatory neural signals are cycling with a consistent relative phase angle. So lots of people have heard of the snappy phrase, cells that fire together, wire together. This is the same thing, more or less, at the large population scale. Populations that are temporally correlated with one another, and that is synchronized, are thought to be communicating with one another. There's a dialogue between them. This is how brain areas might be communicating. And so, again, the local theta gamma phase amplitude coupling might be a signature of the storage of, say, visual objects in the sensory processing areas or features of objects, whereas the prefrontal cortex active during working memory maintenance might be like this fulcrum that reaches back and controls and organizes and coordinates those activities, providing more top-down and monitoring type function. And it might be able to do that long distance control via phase synchronization. There's some evidence for this, for example, in this recent MEG study, where you see frontal temporal phase synchronization at high frequencies, around 8 Hertz, during the maintenance of complex real-world objects in working memory. So this is all to get to our research question for older people. The question is, do age-related working memory deficits that have been observed for decades result from impairments in these rhythmic mechanisms indexed by phase amplitude coupling and phase synchronization? And so our predictions are younger people are going to have intact rhythmic mechanisms of these two sorts, and the older folks, they're going to be attenuated or disrupted in some fashion. So we ran this classic delayed match to sample task with real-world high-fidelity objects. So here's how it comes in. You fixate a central point. You're presented with this real-world natural object. It goes away for a 3-second delay period. You're presented with another object, and you just tell us whether it's the same as the original or different with a button press. That's the memory trial, and the time of interest, of course, is during this delay period. Then there's a control trial to control attention, vigilance, arousal. This is a slick design that we're borrowing from other researchers. So this trial is identical to the memory trial, except at probe, your focus is not to remember the target, not to store the target, but to perform a task at probe, which is just to tell us whether this grading stimulus is tilted left or right from the vertical. And it's matched. It's a block design, and the blocks are matched on perceptual and attentional demands. This allows us to better isolate the neural processes related to memory maintenance within each age group, and it reduces effects related to that are stimulus related or non-memory related. So this is like a really slick technique when you compare the control and memory trials during the delay period to really isolate that working memory neural process. Well, behaviorally, 42 younger folks, 42 older people, we find what they found for 3 decades, which was nice to replicate, that older people are slower and less accurate at performing the delayed match to sample task. And this is just consistent with literally 3 decades of work showing working memory behavioral reductions in older people relative to younger people. So while participants were performing this working memory task, we recorded their EEG, and offline we calculated phase amplitude coupling for a range of phase frequencies and a range of amplitude frequencies between the memory and the control trials during the delay period on the task. And then we used cluster-based permutation statistics to identify memory-specific spatial spectral frequency clusters for each age group. And if we do that, here's what we find. There's a significant cluster of theta gamma phase amplitude coupling in left temporal electrodes over here for younger people. This was true for the 8 Hertz center phase frequency and 30 Hertz center amplitude frequency, whereas no clusters were identified for these 65 and older individuals. So based on this, our preliminary interpretation was that the working memory behavior problems appear to be associated with these reductions in theta gamma phase amplitude coupling in the temporal cortex, which are our inverse source model, which we don't make strong claims about the neural origins of these effects based on inverse modeling, of course, but it is suggestive. It kind of sharpens up our reasoning about where these the source of this effect might derive from to help inform like microstimulation, intracranial work in humans and animals in the future. And this, of course, this is consistent with previous intracranial work that has also suggested the importance of theta gamma phase amplitude coupling in working memory maintenance in humans, also in temporal, medial temporal areas. So we found that the local phase amplitude coupling deficit in temporal cortex arose because of deficient prefrontal control marked by reduced theta phase synchronization between frontal and temporal regions. So here in this analysis, we take the location and the frequency that was identified in that PAC analysis, we seed it, and we calculate this measure of phase-based connectivity. It's the name of the measure is called phase locking value across all the source points in source space. This is our proxy for the cortical surface space. So we can actually move the data from the scalp to the cortical surface, doing some fancy math. And what we find is a cluster of phase locking values in frontal regions, meaning that there's a stream of phase-based connectivity between the frontal and the left temporal regions during the maintenance period on this working memory task. That's present for young folks, but it was not identifiable in older people. So these synchronization schemes, theta gamma phase amplitude coupling and theta phase synchronization, these served as our targets for neuromodulation in order to improve memory function. And this is just to show that this connectivity results consistent with a recent MEG paper in humans doing a similar task. I want to stress that a lot of the work to date in the field of non-invasive brain stimulation or non-invasive neuromodulation isn't as rooted in fundamental neuroscience as the approach that we try to take here. So we developed a personalized HD TACS protocol to rescue is a strong word, but to improve theta phase synchronization in the frontal temporal cortex. And this protocol was exploiting this high resolution approach of TACS, like I mentioned before, is guided by electric field modeling. So we're using these much smaller electrodes, better spatial resolution. It's guided by modeling. We target the frontal and left temporal regions. The frequency of synchronization was individually determined likelihood of entraining these oscillations. So this involved calculating each person's frontal temporal theta phase synchronization during a pre-stimulation session. So everyone actually comes into this experiment before they start the proper experiment. We do a pre-experiment session with them where they do an abbreviated version of our task. We calculate source level frontal temporal theta phase synchronization. We find the peak frequency for you. Maybe yours is 7.2 hertz, so we'll round down to 7 hertz. Maybe someone else is 4.8, so we'll round up to 5, so with a half a hertz resolution. And that will be the frequency of the stimulation that you get injected into these regions. So it's a frequency personalized HD TACS approach. We also applied an in-phase multifocal design. So in-phase means that there's a 0 degrees phase offset between the currents being injected into the frontal and temporal regions. So the beauty of TACS is that we can potentially manipulate these neural timing mechanisms that, the brain is a very fast place, and these temporal features of brain activities within and between regions are important. And so here with this tool, it's spectacular. We can actually manipulate the timing of these neural activities across distant regions. And so we set up this protocol to induce 0 phase lag connectivity. So this is called an in-phase or a synchronizing procedure. So we're going to push the current in and pull it out with a 0 degrees phase offset between these regions to try to facilitate the neural integration between these regions. And we've previously shown, I'm showing this PNAS paper from when I first got to BU, we showed that by changing the timing of the TACS applied across distant brain regions, that we can both up and down regulate phase-based connectivity between those target regions. So this looks like a promising approach. This is something that's done. We're certainly not the first ones to attempt this, but it's especially novel when you can target more than one region at the same time. So the study was double-blind. Participants are blind, and the experimenter is blind. It's sham-controlled, and it's within subjects. So everyone comes in and does an active day and a sham day. The order of stimulations counterbalanced across subjects. On the sham day, it's identical to the active stimulation day, except stimulation just ramps up at the beginning and the end of this 25-minute period to simulate the itching, tingling, and poking sensations that people often endorse on active days. There have been no adverse effects in the thousands of people I've recorded with this tool since 2010. Outside that minimal like itching, tingling, poking, warming, and in rare instances, people feel fatigued and a little like disoriented or headachy. But that's pretty rare. It's quite tolerable. The safety profile is very attractive, and it's one of the great advantages of this technique over other techniques. The stimulation period is 25 minutes while people are performing the task. They continue to perform the task for another 50 minutes while stimulation is off. And that post-stimulation period is our period of interest for where we do data analysis on behavior and neural activity. The stimulation period, EEG, is confounded with the TACS artifact, but we're working on trying to get that out. So we found that the simultaneous in-phase entrainment of both frontal and temporal regions at personalized theta frequencies restored the intrinsic frontal temporal theta phase synchronization and recovered the deficient theta gamma phase amplitude coupling in temporal cortex. So what I was showing you before were actually the sham data from the younger people and the older people. And the new piece here is the active older condition in all of these panels, where you can see the brains of these older people on brain stimulation look quite similar to the brains of these 20-somethings, 40 years younger. So the end result was, if we just changed neural activity, that would have been interesting, but behavior also came along for the ride. So the end result was this improvement in working memory performance in older adults, as well as enhanced phase amplitude coupling behavior relationships. They were stronger. So the stronger the coupling, the better the working memory accuracy. I want to point out on this reaction time and accuracy plots, reaction time did not change, but accuracy did. So this was notable because it didn't appear as if the stimulation was simply inducing a speed accuracy trade-off effect, where we're making you better, but also slowing you down. So we can make you better without slowing you down. And then every one of these data points in this scatter is an individual subject. And what it's showing is before stimulation for older people only, their theta gamma phase amplitude coupling was not predictive of later end of the trial behavioral success. But after stimulation, we could, in a sense, imbue this neural activity with functional significance, being predictive of later behavioral success. Also, even though the neuromodulation was performed for only 25 minutes, the improvements in memory function were sustained for at least 50 minutes, suggesting that the protocol produced neuroplastic effects, outlasting the modulation period. So another experiment in healthy younger people, 18, small sample, so it's preliminary. We did everything the same for these healthy younger people as we did in the previous design, except they received anti-phase stimulation or desynchronizing stimulation. So now the phase offset between these two regions is anti-phase, is 180 degrees phase offset, with the idea of trying to interfere and destructively interfere with this stream of brain connectivity that we're seeing is important for working memory behavior. And indeed, we're able to impair both the speed and accuracy on this working memory task by applying the anti-phase to HD-TACS. So this is nice because, I mean, again, it speaks to the importance of timing in the brain and connectivity. And it's also something we were able to do some years ago in a time estimation learning task. So we found that targeting phase-based connectivity across the medial lateral circuit in the frontal cortex, we could bidirectionally change adaptive behavior and learning in this time estimation task in healthy young adults. So again, we can upregulate or downregulate behavior based on the phase angle of the alternating current that we're pushing and pulling through the brain. We recently, we were curious, like, hey, are we the only ones seeing these effects? What does it look like in the broader field? So at the meta-analytic level. And so we recently conducted, the paper is accepted now, but it's not in press quite yet, conducted the largest meta-analysis of all TACS studies. We have over, I think, 102 TACS studies, looking at TACS effects on various forms of cognition. And we found that the effect holds up across the TACS literature. So examining these 102 studies, we found that the anti-phase stimulation generally disrupts, while the in-phase stimulation generally improves cognitive performance. And again, this is across a large number of different studies: working memory, attention, executive function. So I think one possible application here, or something to explore, would be with respect to PTSD. So, right, this property of neuromodulation, it could prove useful in pathologies where there's an overactive memory process that needs to be downregulated, like in PTSD. But indeed, for any situation where there's overactive connectivity that needs to be downregulated. Importantly, the precision neuromodulation approach in which the HD-TACS is performed at personalized data frequencies was found to be essential, at least for this task, for boosting this type of visual working memory. So we ran another control experiment. We wanted to reproduce the principal behavioral findings and extend them. And so the control experiments were one of them was with a fixed theta frequency for all participants to assess frequency specificity, which did not produce any improvements in memory function in the older adults, in contrast to the personalized approach, which produced robust improvements. So here, reaction times on the left, accuracies on the right. The gray is the sham baseline. The blue is the replication of what we saw in experiment 1 with frontal temporal in phase tuned or personalized stimulation with this selective boost to working memory accuracy. Green is the frequency specificity test, which we found a uniform 8 Hertz TACS waveform was not sufficient. We needed to have it personalized in order to see these gains, although we do note like a qualitative increase relative to sham. And then the yellow and red here are the frequency specificity tests, where we went in and everything is the same, except now we're just stimulating the frontal region alone or the temporal region alone. And it's all within subjects, older people coming in for 5 separate test days, double-blind, sham-controlled, and randomized. And so we see evidence here of spectral and spatial specificity. It's especially noteworthy to point out because I do a lot of brain stimulation research and I read a lot of these papers, and the gold standard really for testing spatial specificity would be targeting a location outside the location of interest, maybe motor cortex in this case, some neutral like kind of region where you don't have a theory that would be an implicated region. And frequency wise, you go to a neighboring frequency band. So we're really focusing on theta here for this study. So maybe a little lower to delta or a little higher to alpha. But what's so rigorous here is that we're actually within the frequency range of interest and within the brain regions of interest. So it really goes to show you just how specific you need to be. You need both regions modulated concurrently. Again, that speaks to the timing nature of what we think we're really isolating and augmenting here. And you also need to be personalized in the theta band. You can't just give a uniform theta frequency stimulation. Again, there are some caveats. This might be specific to this task or specific to kind of processes we're manipulating. So the upshot is that age appears to impair, but this form of HD-TACS could potentially rescue these really well-studied signatures of brain information transmission and plasticity, theta gamma phase amplitude coupling and theta phase synchronization, most well-studied in the animal literature, systems neuroscience, as well as age impairing and HD-TACS improving working memory performance accuracy. And then we also find that these components of the mechanisms that we're changing, or components of the tool that we're using, are really important: space, frequency, and phase. So our results are consistent with cognitive theories that propose cognitive decline results from cortical disconnection from structural and functional connectivity changes, at least in part. Results are also consistent with theories that propose that link prefrontal cortex and aging as being an important to understand that the prefrontal cortex has this particular vulnerability in older people, and that is consistent with what we're showing, that older people really struggle to use the prefrontal cortex to synchronize and drive subsequent coupling for memory performance. And even at a higher level, we think it's really exciting that we may have a tool now to non-invasively intervene in the temporal coupling of these distant rhythmic activities in the brain to either optimize or impede cross-area communication that's important for memory processing and behavior. Of course, there's more than just working memory. There's another dominant memory system, the elephant in the room, the long-term memory system that we're also interested in, our capacity, unlimited, this profligate capacity like system. And within this dual store framework, there's many frameworks, but this classic dual store framework where there's a working memory that's distinct from a long-term memory, we know that decades of research shows that these different memory systems can be indexed separately by the primacy and the recency serial position curve effects. Right, so if I read you a list of 20 words and I ask you to freely recall as many of those words on the list that you can, you tend to be quite strong for remembering words early on the list, later on the list, but do a poorer job on recalling words in the middle of the list. And this classic serial position curve effect, this function is what you get. So there's an advantage for early words that's known as the primacy effect linked to long-term memory, and an advantage for the latter words in the list deemed recency related to working memory. And the linking of long-term memory with primacy and working memory with recency is supported by almost a century, really large experimental psychology and neuropsychology literature. So we know that the dorsolateral prefrontal cortex and inferior parietal lobule are big players in the functional neuroanatomy of memory. With DLPFC, it's not that clean, but there's a drift of evidence that that's primarily linked to primacy in many different tasks, and the IPL, the inferior parietal lobule, drifting toward being important for subserving recency. What we don't know is whether there are distinct rhythmic brain activity patterns in these regions that govern the different memory capacities. So the idea is, like, if there are unique rhythmic mechanisms in these spatially distinct brain regions, if those can be identified, then these brain rhythms could potentially be independently and non-invasively targeted and manipulated with techniques like HDT-ACS to selectively improve memory function in older people, which could be especially important if you're just suffering from a working memory problem or just a long-term memory problem. Having that kind of flexibility and selectivity in a tool, you can imagine having great clinical utility. So over a series, so I'm sorry I didn't point out, but that first experiment study, that collection of experiments, was headed up by a PhD student called John Nguyen in my lab. This one is headed up by another PhD student in the lab, Shrey Grover. So over a series of randomized, double-blind, sham control experiments in 150 older aged adults, 65 to 88 years, we, meaning mainly Shrey, tested whether modulating theta rhythms in the parietal cortex could improve, in this time, auditory verbal working memory function, the recency effect, whereas modulating gamma rhythms in the prefrontal cortex could improve auditory verbal long-term memory function, the primacy effect, in older people. To do this, we designed two novel HDT-ACS protocols by using current flow modeling and previous literature. And we wanted to cause long-lasting effects as best we could. So we stimulated repeatedly, once each day for 4 consecutive days, and tested memory performance out 1 month after the intervention. So here's what we found. So what you're seeing here, there's a lot of stuff going on. Experiment 1's on the left, experiment 2's on the right, shams are on the top, then on the left, we'll focus on experiment 1 here. Sham is gray, the DLPFC gamma stimulation is blue, the IPL theta stimulation is orange. As you move from left to right, you see primacy, middle 1, middle 2, middle 3 recency. And then the color coding is the lightest color is baseline day. And then as you get warmer in color, you move through the intervention, day 1, day 2, day 3, day 4, and then 1 month post. And what we're seeing is that the modulation of the low frequency, but not the high frequency activity in parietal cortex preferentially improved working memory recency on day 3, day 4, and 1 month after the intervention, whereas modulation of high frequency but not low frequency activity in the prefrontal cortex preferentially improved long-term memory primacy on days 2, 3, and 4, and 1 month after the intervention. We sought to confirm the location and frequency specificity of our hypotheses. So we ran a second experiment here, experiment 2, where you can see that we just switched the entrainment frequencies in the 2 regions to examine the effect of gamma entrainment in parietal cortex and theta entrainment in prefrontal cortex on memory function, which, as you can tell, resulted in no changes to memory performance. So the upshot here is that not just location matters in the brain. We've known that since Ramon y Cajal, but obviously we've also known it more definitively from all the neuropsychology as well as the TMS work, that DLPFC for primacy, for example, and IPL for recency are causally involved. But that frequency, the big novel basic science insight here is the frequency component, in that the brain, nature cuts the brain along these kind of frequency channels of information processing, not just in terms of space. So we're finding a double dissociation between, on this dual store framework, between working memory and long-term memory on one hand, and between space and frequency of the mechanisms of these memory systems on the other hand. So that was pretty cool. I think Nature had a review on this, and it was entitled a location frequency to remember, which I thought was pretty clever. So we're also finding that the rate of the memory improvements over this 4-day period predicted the size of the memory benefits 1 month later. And in addition, the individuals with low baseline cognitive functioning, as measured by the MoCA, the Montreal Cognitive Assessment, experienced larger and more enduring memory improvements. So again, the rate of memory improvement, so the speed at which your memory benefits are increasing over the 4-day intervention, is that rate is predictive of how big your memory benefits will be 1 month later. So potentially you can imagine in a clinical setting that might be helpful to know how well you respond to the intervention right now during the intervention will predict how well the outcomes might be for you months later. And then in addition, the procedure appears to help people with the in the poorest, the poor cognitive functioning people the most, in terms of predicting the rate at which they improve during the intervention and how big the benefits will be 1 month later. So this also bodes well potentially for porting this into a proper clinical trial with people with MCI and mild, moderate Alzheimer's disease, as obviously their memory deficits are going to be larger than these healthy controls. But we had enough variability to see these relationships, which I think are encouraging for clinical research. At the meta-analytic level, we also looked at TACS effects on cognitive function, particularly in older adults, during and after TACS. During TACS, the online effects, and after the procedure, how long the effects last, which both actually became even more significant after we removed a few outliers, but were significant before outlier analysis as well. So we're seeing small to medium-sized effects in the literature to date on older people using TACS to improve various cognitive functions, working memory, executive functions, attention, so on. So certainly more research is needed. This is still a small batch of studies that we have to date, but it looks encouraging for evidence for cognitive enhancement using TACS in older people. Generally, looking forward, we're cautiously optimistic. We're interested in trying to continue to use these insights from systems and cognitive neuroscience to build better neuromodulation protocols, to selectively change aspects of brain networks and improve cognitive function in rapid ways and long-lasting ways, and hopefully in the service of helping people with serious brain disorders. So that's what I had. Thank you very much. Happy to take your questions.
Rob, thank you so much. This was a really stimulating, enlightening talk. There are a number of questions in the chat. Some of them were asked early on, and I think you kind of answered some of them. So, but I'm going to go through them anyway. So is there any risk of causing seizures with, when you're synchronizing brain activity? That's
There's been no reports of seizure induction in the literature to date. So unlike RTMS, where that risk is real, with these neuromodulatory approaches. So this is a good point, because this brings, so the short answer is no. And one of the reasons we think that's the case is because these are neuromodulatory approaches as in contrast to neurostimulation approaches. So that means that with a tool like TMS, you can recruit neural firing right away. It's an order of magnitude larger, the induced field that you put into the brain. These are such with TDCS, TACS, and random TRNS, oscillatory direct current, this whole family of electrical transcranial methods. They induce an electric field that is, say, 0.3 volts per meter. So not enough to make a cell fire on its own. And in fact, that's what the intracranial work in animal models shows, that it doesn't induce de novo firing. You need the system kind of active and in order to modulate activity. So you're you're increasing kind of ionic gradients to a point where you increase the likelihood of making a cell fire. You're kind of nudging these networks into a different mode of functioning, but it's not creating, not making action potentials increase in rate like right away, like with TMS. So it's just that's why it's regarded as like low intensity and modulatory. And I've kind of mixed up the terms a little, but I'm trying to more and more in my publications use terms like neuromodulation instead of stimulation or neurostimulation, just to help make that distinction.
We're all getting more sophisticated and more carefully defining our terminology. So another question that I think a lot of clinicians will be interested in your answer about is about commercially available devices for neurohacking. I'm sure you get asked about this a lot. Again, my sense is that you have kind of answered this by clarifying how detailed and specific the application needs to be in terms of frequency and location. But what are your thoughts about this?
My thoughts are, I think it's premature to have these spin-offs into the commercial market because there's so little that we know. And that's kind of like, hey, that's my bias. I've been trained as a basic scientist, so of course I'm going to say that. We need more research. But I do really believe that. And I'll tell you for one reason, we make mistakes in the lab all the time. And when you're piloting and figuring something out, and the brain is this like delicate machine. And you could have unintended consequences. I mean, this is a whole other conversation about like medical ethics. And there have been cases where we don't have full exhaustive understanding of these circuits and networks that we're manipulating. And so it's actually really nice to have a tool that has such a strong safety profile. But there have been cases where we're stimulating a region and we're seeing improvement. And then in another case, someone will come in, maybe someone new to the lab, and they'll place the electrodes not in the correct place. And we'll see effects flip, where you're creating a deficit instead of a benefit. Mm-hmm. And we this is not published, but in that specific moment, based on the EEG data, it looked like we were on the other side of an electrical dipole. And that might have something to do with it. So it's just generally don't know enough, would feel very uneasy about spinning this off into like a wearable device. But I know that's not stopping industry, and that there's a big appetite for neurostimulation and these types of devices, and they're already there. So kind of need to start having the hard conversations about ethics and it's also, these are devices that are pretty easy to make on your own, right? Like 9-volt batteries, some tin foil, some wires. We already know there's home hobbyists posting YouTube videos. Someone could get hurt.
Absolutely.
These are not like toys.
So, yeah, I thank you for your perspective on that.
Yeah.
Some other questions here. One is, what did the participants report? The ones who had improvement in working memory measures, what did they report subjectively?
Yeah, it's a great question. I get this so often, and honestly, we have to do a better job characterizing people's subjective experiences. I don't know if we're biased because of our training, we're just after these objective measures more and more, but getting that subjective profile and listening to people, that would be just really important to do. Now, we do questionnaires and we do this visual analog scale. We have something, but it should be richer so we can really capture the most of it. I have anecdotal stories to tell along these lines. But systematically at the population level, people are at chance in terms of guessing what condition they're in generally. And the way they self-report on questions about sensation under the electrodes, their mood, their attention, are not significantly different across the placebo and the active conditions. So it doesn't look like people can like unblind themselves. On the other hand, there are like notable exceptions where we've had personal experiences with individuals that are like, wow, I feel like, and they describe what it's like to have this expanded memory or this increased visual acuity. Thinking in particular, I've had some dramatic effects of, in this case, it was TDCS over visual cortex while individuals performed the Snellen acuity chart. That's the standard eye doctor chart where we read the letters. And some people just unblinded themselves because the stimulation had such an enhancement in their acuity, which was what we were trying to do. But some people just, they could read like 7 more letters or some, whatever it was, 4, 7, some considerable number of more letters. So they were describing their experience as the letters being more crisp and less fuzzy. And in those cases, it was pretty interesting. And there was like an unblinding of participants. But typically, I think a lot of the effects that we're observing, although systematic at the population scale, they're relatively small still. I mean, so that the data I showed in the first experiment, there was an accuracy change of 10% on average. And so that's the kind of thing that I think could fly below the radar and not have people become consciously aware of.
Well, that's what I was going to ask as a follow-up, whether the people who reported this wow kind of experience where they unblinded, themselves, whether they had more profound, effects, more dramatic, actual measurable effects.
They are the ones. Yeah, they typically are the ones that have these really
so
strong
maybe
positive response.
Yeah. So, okay, let me go on to some other questions here, rather than just having a conversation, just the two of us. One person asked, working memory deficits are so big in schizophrenia. How has your work in that area gone?
I haven't, the work in schizophrenia is mainly from my graduate school days. And, and that's where we used TDCS, direct current stimulation, and targeted executive functions. And these days, my work in schizophrenia are with Macheri Keshafan, who I see is also here. We have a number of ongoing projects. So it's really time and investigation will tell. We haven't gotten to, I think it's a real low-hanging fruit. Bring in the patients, let's evaluate their visual working memory and apply this procedure and see if we can learn something about pathophysiology, underlying visual working memory deficits, and how those relate to symptoms. And we know that it's actually the cognitive symptoms that are the highest predictor of poor functional outcome in patients, accounting for their crippling disability as opposed to the positive and negative symptoms. So hitting those cognitive symptoms and having a tool to address that would be really extraordinary. Sorry, I don't have too much to say on that, really taking the memory research findings into the Alzheimer's direction, but it could be taken in so many directions. As far as the executive functions that we did some nice work with Sohee Park, graciously made her schizophrenia population available to me in graduate school. And we did some work with Sohee Park at Vanderbilt, where I was a student with Jeff Woodman as my mentor. And we showed that we could use TDCS over medial frontal cortex, which is thought to be a real major hub in the cognitive control network, and see changes in low frequency oscillations and that a full TDCS treatment for 20 minutes. And as a result, see a behavioral marker of anterior cingulate cortex functioning of adaptive behavioral control measured in posterior reaction time slowing in schizophrenia patients who have known for decades to have, well, there is some controversy, but the drift of the study suggests there's a reaction time slowing deficit in patients, and we're able to normalize that. So presumably enhancing their ability to adapt after they make an error on a task with that one brief, one shot of TDCS over the medial frontal region. That was a paper with Sohee Park and Jeff Woodman in 2015. So that would be really exciting to follow up on as well.
Yeah. So much to do. So one person asked a couple of questions. I'm just going to read them. Did you account for the theta phase lag or lead between frontal and temporal cortices when you were conducting theta TACS? And then, great talk. Do you have experience or have you heard of performing TACS stimulation simultaneously across two frequencies, such as gamma and theta band, to improve theta gamma coupling?
Yeah, exactly. I mean, that's exactly where we're at in the lab. We're interested in that in cross frequency coupling. So, with the device we have, anything we can program into MATLAB, we can deliver as a stimulating waveform. And I'd be very interested in doing frequency coupled waveforms, since we have frequency coupled neurosignatures that are prominent in various aspects of cognition. So yeah, and it's like there's some theoretical insights there that can really guide us. There was a nice PLOS biology paper, I believe it was, that showed in that regime of theta gamma coupling. So the idea, like very cartoonish, is that gamma may be the objects you're storing in your working memory. The cycles of gamma are the objects you're storing in your working memory. So 3 gamma cycles, 3 objects, 4 gamma cycles, 4 objects. And however many you can fit on one theta cycle, however many of those gamma bursts you can put on one theta cycle, well, that's your working memory capacity limit. And so how can you get more in on a theta cycle? Maybe you can entrain the brain to causally slow down the theta cycle. So this is beautiful PLOS biology paper, I think it was, that showed that TACS over right parietal cortex in a visual working memory task, as you slow down the theta frequency, you increase visual working memory behavior capacity. I think it was measured with K capacity. That was really cool, aligns with Lisman's theory of the theta gamma coupled oscillators for working memory performance. So whether you're doing it like a single oscillation like that, just slowing down theta, or where you're coupling the frequencies, really interesting. That's something we want to do in the future. Your first question, pertain to the neural timing mechanism. So this is pretty interesting. We're not the first to show this, but first, taking a step back, there's zero phase lag connectivity that's been shown in animal and human research to be behaviorally meaningful. I have a discussion of this in that 2017 PNAS paper. And so it's not surprising that with zero phase lag, we can see corresponding changes in the behavior that's key to that zero phase lag. But it's still interesting that it suggests we don't have to do this meticulous accounting of axonal conductance delays across long distances in order to have success in using this technique. On the other hand, that is something we're paying attention to now. I'm curious because there's some new modeling work with this multifocal TACS, dual region TACS. And in the modeled neurons, it shows that there is some kind of optimal, like, sweet spot if you account for these small conductance delays. And then it drops off very rapidly where it doesn't help at all. So maybe with very small conductance delays, so maybe there's more bang for our buck that we can get out of this tool if we do that accounting. But for the work we've done to date, we haven't. It's been 0 phase lag connectivity that we've targeted. So that's a great question. That's another one, kind of frontiers topic.
A couple of other quick ones. One is, how durable are the pro-cognitive effects in the elderlies in terms of days or weeks? And then the second one is, what about ADHD?
Well, ADHD, I have a collaborator. Well, I think ADHD it's definitely the tools that we created in 2017 that can increase. I showed you little pieces of some of those findings throughout this talk. It was that 2017 paper where we were able to go in with theta TACS over the medial lateral circular frontal cortex, which is for decades known to be involved in adaptive behavioral control, this stream of theta coherence, and we could push it up or push it down based on whether we were synchronizing or desynchronizing with the TACS. And we can see those corresponding bidirectional changes in behavior, making people faster at learning or slower at learning, or making them adapt more efficiently after they make an error on a task or adapt less efficiently. That was pretty remarkable. So that would be where I would put my bet for the protocol to apply for in the ADHD case. So I think that's another piece of low-hanging fruit. Since ADHD, we obviously have attentional problems and executive control. I think ADHD, when I think of ADHD, I don't think of attention as much as I think of executive function problems. And that might be a nice way to start to chip away at that. Yeah, I think it's potentially addressable with these types of tools. It'd be very interesting to look into that. We don't know how durable the effects are. So right now, in the first experiment, we looked just across the first 50 minutes after that 25-minute dose of stimulation. But we don't know. It's part of an aim to a grant I've submitted to look at the duration of the effects more systematically. And in the last experiment study I showed, where we looked at 1 month later, it looked like the effects sustained for that 1 month. But they came down a little. If you could remember the graph, they kind of came down a little relative to the fourth and last day of the intervention. So they probably come back down to baseline levels, presumably at some point, but it would be nice to track that out and to get more hard data on exactly the time course of the effect. So I actually think that's like an entire grant all by itself, just the duration of the effects of a lot of these
So somebody asked about brain-derived neurotropic factor, whether you've measured that.
I haven't measured it. The closest thing I've seen is work with pharmacological challenge day studies in humans using agonists and antagonists related to spike timing dependent plasticity, and how that can be affected by stimulation, suggesting the stimulation is having its behavioral effects because of spike timing dependence. But I'm not sure about BDNF. Actually, I don't have much to add on that. That's a good question.
Okay, and then I just have one final question. Well, two final questions. One is, when you noted that obviously you're doing personalized treatment and you're measuring frequencies that you're tuning to, what do you make of the variation from person to person in terms of the different frequencies? I mean, that's a really general
Yeah.
question, but I mean, is there any, you probably haven't studied it, but is there any pattern to what
Well,
sense of what it says about the individuals or?
no, that's a really interesting point. Well, I mean, we know there are like spectral rotations across age groups in the alpha band, where older people's alpha is slower than younger people's alpha. But
Okay,
that's
so
not really what you're asking. You're asking like individuals like within the same age group, why the variability? I mean, I guess I could have like hand wavy comments about neural noise and
individuality, genetics. Yeah,
right,
yeah.
like I mean, skull thickness, like cortical geometry. I mean, it could be like physical. I don't think it's at like it's related to any like fundamental computational process. I think that's going to be like species specific. But when you get up at this level of like phenomenology, this flux of lots of complicated interfering factors, individual differences, there's a lot. There's so much of like interfering things that could give rise to changes at the surface, a complex system. But I think, for example, it might be that there's a one-to-one correspondence between particular oscillatory frequencies and particular like computational subroutines. That's fixed and invariant across the species at a really low, like basic level. But then when you get up, in the normal case, I'm not sure what that would look like necessarily at pathological extremes, people with mental illness. But it's not clear. It's not clear to me why the variability. That's something we haven't really explored much until we've got into personalizing, and we're wondering about the same thing. So more on that in the future, Barbara. So I don't have a good.
Okay, the future. That's my last question. What do you think about the future? What do you foresee, 10 years out? What are your hopes?
Yeah, it's my hope. Oh, that's very easy to answer. So I think
Good.
I foresee lots of like technological advancements, but they also they actually it might surprise you to know that they actually worry me in the sense of having all this technical resolution. I mean, we have amazing tools at our disposal, but at the same time, I have to put on my basic science hat and be a curmudgeon a little and say, where's the conceptual resolution? Where's the theoretical muscularity in all this? I feel like we have tons of tools, and everyone can use them, and they provide this pattern of results that it's just like difficult to interpret, or it's just shallow and thin. The understanding is thin, not just about mechanisms underlying these tools, but about mechanisms of the brain in the normal case. So I think I'd love to see better like theory building, computational neuroscience brought to bear in conjunction with these fancy neuroscience toys that we have more and more. So I'd imagine the future, that history's any guide, we're going to see a lot more technological innovation, but my hope is that it be paired with conceptual or theoretical innovations.
I love that. That's, your deep thinking. We really appreciate it. And hope you'll come back again and update us.
My pleasure. I hope I can join you at a meeting in person in the future.
Oh yeah, we would love to have you.
Thank you
Thank
so
you
much.
very much.
Thank you, Rob. Thank you everyone for joining us. This will be available on our YouTube in a couple of days, so check back on that. If you'd like to rewatch or share it with anyone, feel free. Just want to thank Dr. Reinhart one more time for a great talk, and everyone have a great night.